Hello, everyone, and welcome to this edition of the CINECA webinar series, where Will Rayner and Anshika Chowdhury from Helmholtz Munich will present federated analysis for polygenic risk score calculations. My name is Marta Llorell Linares, and I'm involved in the CINECA project on behalf of MBLBI. I would like to make you aware that this webinar is being recorded, and this recording will be available on the CINECA website and YouTube channels. At the end of the session, we have reserved some time for questions from the audience. So if you have any questions, please write them on the Zoom question and answer panel, as you can see in your Zoom app. I'll briefly introduce the CINECA project. CINECA stands for Common Infrastructure for National Cohorts in Europe, Canada, and Africa. And it's a 4-year project funded by the European Union's Horizon 2020 Research and Innovation Programme and the Canadian Institute of Health Research. CINECA's vision is to accelerate disease research and improve health by facilitating transcontinental human data exchange. And to achieve this, CINECA will address several challenges. So the first one is federated data discovery. So how, as a researcher, you find cohort data of interest. This could be for a specific genotype or phenotype, for example. And then once you find the data, you will need access to it. So CINECA is working on an interoperable authentication and authorization infrastructure. And once you find the data and you want to ask research questions across cohorts, there will be a need for a harmonized cohort-level metadata. The ultimate aim of CINECA is to enable federated data analysis for research and healthcare applications. So CINECA is working with a set of demonstrators in both research and clinical settings. And on top of all this, there's a need for a transnational ELSI framework to address the ethical, legal, and societal issues. CINECA also has an outreach and training program to raise awareness and capacity building in these areas. So I'll briefly introduce today's presenters. Will Rayner is the head of the Data and Analytics Group at the Institute of Translational Genomics in the Computational Health Department at Helmholtz Munich. He's interested in all aspects of data management and data privacy and has been leading the CINECA Polygenic Risk Score use case. Anshika Chowdhury is a data informatician at the Institute of Translational Genomics at Helmholtz Munich. She has been working on the development of the workflows for the polygenic risk score use case and analysis of the EQTL catalog on the datasets at Helmholtz Munich. And now I will hand over to Will.
So I'm going to give a brief sort of introduction to the reason behind our work on the federated analysis, and sort of, I think, maybe I don't need to state this, but sort of what is it? Why are we doing it? So what is it in that we are actually running analyses on multiple sort of genomic data sets that reside in different computer systems. And we're doing this as if we actually had access to all of them locally. This has been something that's been going on for a very long time, and there are very large consortia that have been doing this kind of methodology, in that we end up with multiple cohorts. We ask them to run an analysis for us. We present them with an analysis plan. They generate a set of data for us, normally summary statistics, which we then take. We then concentrate that centrally and analyze it as a meta-analysis in this kind of diagram. And this is something that's been going on manually for quite some time, and it has been very successful. It's delivered very large numbers of associated loci with genome-wide association studies, but it's really slow. And the really big ones, like the latest giant set, has taken sort of somewhere around 5 years to actually be able to run all of this and get all the data gathered and cleaned. And so this is something that we think that needs to change and to speed up. And why? Because there are all these huge cohorts that are being generated around the world. And this was a slide from the original CINECA application from 2018. And these numbers will be well out of date now, but people are generating larger and larger data sets, both for research, but also biobank, and also using sort of the health data as well. And I think part of this for me is this is the European health data space, which is one of multiple data spaces that's being planned by the European Union. And the idea is that individuals control access to their health data, and they can then port it with them across countries within the EU. And this will allow people to have better control, but also it's built in that there's going to be the possibility of using this data for secondary use, i.e. research. And the way this is looking to be structured is that there will be kind of a, say, similar sort of layout to what we've been proposing in the Seneca project, with metadata, dataset catalogs, people searching for the data, authorization, and then people logging in. And these are some slides from the EU proposing how they're going to do this for certain secondary use, but it's also going to be set up sort of EU-wide. So there will be one or more health data access bodies per country in the EU, and they will all have these trusted research environments that allow you to access the data. And they're talking about how they set up the data, and they have this kind of visual diagram of the potential that, you know, how you request the data for the metadata catalog, how the application is handled, and that you then run you bring your code to the data, run it in the secure processing environment, and extract the data. And this is really very nice to see, but part of it is that you're then ending up with multiple data sets in multiple silos in these processing environments, and these need to be integrated. And, you know, their view of this is that, you know, you through this workflow, how you request and access the data, but you get access to the secure processing environment, but actually you need, I reckon that should be secure processing environments, as you need access to more than one data set to be able to combine the data. And so this is, I think, a real strength of Cineca, and a vision that sort of predates all of this. And I'm going to briefly show one slide about the structure. And as a standard EU project, multiple work packages. So we're kind of this work package 4, but we're looking at basically generating research use cases. And our research use case has been to look at genetic data and genetic research. And for this, we've chosen to work with polygenic risk scores, as we felt at the time this was a really useful and interesting area. And I think with that, I shall now pass over to Anshika, who will be able to introduce the work that we've been doing on the polygenic risk scores, and then give a demo of the pipeline that we've put together so far.
Yeah, so I will be continuing with the explanation with regards to the polygenic risk score use case. So a brief introduction of what actually polygenic risk scores are. So a PRS score gives us a single value estimate of an individual's genetic propensity towards a given trait or disease. So it can be a trait like height, or BMI, or some disease like diabetes or cancer or so on. So in principle, it is calculated as the sum of the risk alleles present in an individual weighted by the effect sizes that are derived by association analysis of the same trait from other GWAS studies as such. So of lately, there have been several applications of the PRS scores, where obviously it is used to identify the individual risk for a given or the propensity towards a given trait, but also identify genome-wide interactions with other genes or also with the environment to confer certain causal relationships, and also for population stratification. So as Will already mentioned, we have been working on the work package 4, the specific use case being the computation of polygenic risk scores, where we have been developing a pipeline using Nextflow. So this is an workflow management framework or a language, which is an extension of Groovy, which is again an extension of Java. So we share certain semantics with Groovy as such. The main advantages of using Nextflow are that mainly that the scripts or the pipelines, they are reproducible assets, so you can deploy them in any computing environment, and it allows portability in such way, and also it supports parallel execution of different processes. Obviously, we can include other scripts which we have already been using, for example, certain R scripts or Python or certain tools which have been used extensively for a given data analysis. And one main feature I would say of Nextflow is that it is quite scalable, that once we have a fast prototype, it can be scalable to large pipelines where it can interact with other pipeline in terms of modules, and then this can be shared across various platforms. So these are the main components in a Nextflow script or a project as such. So we have a main script which is executed run. So it basically contains the processes which are to be executed, and they are called from an workflow path as such. So this is very domain specific. So currently, most implementations are in either DSL1 and or 2 as such. So a process here, it contains the main features. One are the input files that can be given from channels or certain values, and then the outputs which are generated from the scripts which are there in a process, and these processes can interact with each other. So in order to run the main script, we also need a configuration file. So once an Nextflow script is executed, the pipeline is executed, it automatically looks for a config file, so which contains the details with regards to the dependencies and the environments needed for running the script as such. So there can be details with regards to the container, which process uses what sort of container, whether it is Singularity or Conman, the parameters required to run that, which executor are used, for example, Slurm or AWS batch or as such, and the cluster options for them. And if we have proper knowledge of our environment as such, then we can also use something like Conda or load certain modules depending on where you want to run the script. And obviously, along with it, we need to give the input and output. So normally these are to be given by the user, or some link has to be provided which can be downloaded on the fly as such. So along with these, the main features of other features of Nextflow are that it provides us with the metrics with regards to the computational resources that are used in each process throughout the pipeline. And as already mentioned, the pipeline can be shared across various domains, and we can also test the scripts step by step, where you can resume the process in case the running of the pipeline fails at some point in time. So you can check the log files which are provided in the file, generated in the file called nextflow.log, and there we can check which process fails at what point in time and what changes can be done, and the pipeline can be resumed from there. And one main final important feature is that we can generate emails with certain metrics that we want and send it across to the responsible people for checking the pipeline execution, and also generate an overall report with regards to the processes that are run, the input and output files that are generated, and the resources that are used in terms of an PDF file or an HTML file. So in the PRS use case, the steps that we mainly implemented are in 3 modules. One module is having a very basic step where if the user provides a VCF file, it is converted into other genotype files which can be read by Plink. And the second module is contains the quality control steps for the target data, again, which is given by the user. So here, the SNPs that have a low genotyping rate, which are out of Hardy-Weinberg equilibrium, or are having a low minor allele frequency or duplicated or unambiguous, they are removed at this point in time. Also, prior to this, if the target data, if it is known not be imputed, we can send the data over or call a module for the imputation of the data as such, and then run the QC steps. The next module is to do the quality control of the base data. So this is similar to that of the quality control steps of the target data. So if an GWAS file is with the summary stats is provided, it is used, or otherwise, and download link is also fine where it is downloaded on the go. And here mismatching SNPs or duplicate and ambiguous SNPs are removed. One step which is to be added is to check for the genome build, or otherwise use something like leftover to check to convert the genome build of the base file as such. So I mean, since the data set that I used was having the same build, I didn't quite focus on implementing this part, but yeah, this is an additional step required for the base quality control steps. And then finally, the input from the base QC and the target QC are given as an input to the PRS computation, which is performed by the software Precise, and this does the clumping, the p-value thresholding, and gives us the PRS scores for each individual in the end. So in case of our use case, the steps needed for running the workflow are that firstly checking out the pipeline to a given computing environment where we want to run it. So the main file along with the config, the required config with the runtime parameters, and also the input and output parameters are given as the input and run. In case of NDSL2 implementation, the main file will first load all the modules and then run each of the processes depending on the workflow script present over there. And as I already mentioned, an imputation check and an association analysis can be performed separately in case if we are not having a base GWAS, or the imputation is not performed in the first place. And while the script is run for each process, and directory is generated inside a directory called work directory, where the intermediate results, the command that is executed, the log of the command, or if there is an error, the error of the command are stored in that directory. So we at any point in time, the directory can be checked to check what command has been performed, or what went wrong in case the script fails. And of course, the status of the pipeline can again be checked in the Nextflow log files. And I have basically implemented and tested the use case on the CINECA UK1 synthetic dataset. So this was derived from the 1000 Genomes project, which has around 2,500 samples and around 84 million SNPs. So the height data was taken from the XML files. I mean, I wrote a simple parser for it to get the height data for all the samples, and then this, along with the height GWAS file, was given as the input for running the PRS pipeline. So currently, the use case, it is implemented on the height data, but obviously other phenotypes and covariates can be given along with given and based depending on the phenotype data as such. So I have a small video to show you how the pipeline is run. So as you can see, yeah, I'm using Visual Studio Code in order to create my project structure, and we have to use it basically as an editor, but yeah, one can use their own preferred editor as such. So here I have my location of my project directory, which is the PRS_demo. So this is present in the HPC cluster here. And when I click on it, it prompts me for my password, which I give as the input. When I have to give it twice because I connected from another server. So yeah. So it so basically the directory is open, and you can see here for any Nextflow project as such, these are the basic required files that are needed for running the pipeline as such. So we have the bin directory, which contains my R scripts or my Python scripts for transforming the data or getting some data sets as such. And this is my conf folder, which contains the configs which is used for running the pipeline. For example, here I'm using a simple config because I'm using just a container for a singularity based container for running my pipeline. And next, there is my input directory, which contains all my input files. So it has all the genotype files and the height trait data for these for these samples as such, and also the summary stats, which was downloaded earlier. And the next is my script files. So basically, I have all my modules present here, namely the base QC, the target QC, GWAS computation, and VCF to link. So what I would run the main file here right now, which is in DSL1. So basically, this is in the form of an monolithic architecture. So all the processes are present here. All the input are already present in the config file. So it takes the inputs and runs the entire processes together. But otherwise, in DSL2, these modules can be called individually as such, and these are loaded on the go in the main script. So I will play this further to show you the main file which is to be run. Yeah, so this is the main file. So basically, I have it DSL1 enabled, and it has all the processes with regards to the QC and the PRS computation. And you can see there is also a results folder as such now, which is empty. So I give this as an input parameter again to the script so that it knows that there is a location where my overall output files have to be present. So I am loading my Conda environment. So this is not for running the pipeline as such. It is just to load Nextflow, because specifically in the case of our cluster here at SMGU, it is that Nextflow is not able to create jobs as such, Slurm jobs, where in case it actually should be able to do it by itself. So in order to run Nextflow from another Slurm job, I created a small environment to just run Nextflow. But otherwise, if it's already present as, then it is not needed. So I am running my main file, but you can see that the location is wrong. So it can't find the script, and you can see that there is a Nextflow log generated automatically here. But now I will give the main, I will give the correct location of the file, and it will generate, it will launch the Nextflow, and it will start generating the outputs, and it will start, basically it will run the processes and start generating the intermediate files and generate the outputs. So here you can see that there is and tag or a runtime name. It's called stupefied_monodes. This is the name given to this runtime as such. So in future, at any point in time, if you want to check the logs or the outputs corresponding to this run as such, they can be checked using that name tag. So you can see that all the processes are running. So basically the QC of the base ran rather quickly because it's a simple file, and the QC of the link files, the genome files also have run. And I kind of fast forwarded to this point in time because the PRS computation takes a little bit more time. So each process, when it runs, it shows the percentage of the run, and then also a small status of whether or not it was able to run completely. So this being a small file that I have given now as input, it runs for around 2 or 3 minutes, and then finally it executes and it shows me that the run was successful. And what you can see in the directory now here is that a work folder is created, which has a set of corresponding folders for each process in the script. So the work, directory here has the command, the input command which was given here, the error, if just in case if there is some error, or the logs of that command, and also the input and the output files of that particular process as such. So if you want to keep track, or if you want to check something, you can go there and check. There is also a nextflow.log file created here. So it gives all the summary stats of the entire script, how it is run from the beginning to the end. So that is also a place to verify how your processes have run. You can see there is an log file still present from the previous run where the location was incorrect. So it just shows that it could not find the file here. So like this, they are like around 10 files, up to 10 files stored for your previous runs. So that is also a place where you can go and check the status of your scripts, or if there are any mistakes that you have done as such. And finally, we coming to the results, we see that there is the set of output files created. It contains the PRS out file, the corresponding bar plots for the p values and the fits, and then also the PRS scores for each individual, and so on. Here you can see that I also was able to generate some files for the GWAS. So it's actually not advised, or it is not mandatory to do so. Just in case, if you want to check, I did it just in case I wanted to check my my files after the QC was performed on the base data. So here these intermediate files were generated. So they are present in the work directory as well, but to have an easy access, I also published them here just in case. And then you also have the cache directory here for the Nextflow in case you want to have a look at it. Yeah, that is pretty much it with regards to the demo. So coming back to the presentation. So this is how the PRS workflow looks from end to end in the context of an federated analysis. So basically, dataset cohorts can be given as the input to the Nextflow, and obviously the main channels provided will be the base data and the target data, and they will be QC'd parallelly in case if the base data is not provided and we have another set of genotype files where an association analysis can be performed. Other modules with regards to the GWAS analysis can be called, or otherwise an imputation server can also be called to perform imputation on the target data. And then the PRS computation is performed, which is given as which can be taken as input for other meta-analysis as such. So in terms of the federated analysis, what we have been doing and plan to do are these things that firstly, like convert the pipeline to DSL2. So this is done, say, up to 99%. There are some minor issues which just happened like yesterday. So yeah, this enables actually modularity, more modularity for the system. So for the pipeline to run. So that is why yeah, this has to be, it is better to have a scripts in DSL2 and then test it on other environments other than Slurm-based environments. And then I have also been working on running pipelines from the EQ2L catalog in the work package on other datasets as such. So this is to have a common framework for all the use cases here for the EQ2L pipelines and also for the PRS as such, in terms of having same similar project structures and having which makes possible interactions between the pipelines much easier. And obviously when we implement the federated analysis, the GA4GH guidelines have to be followed. So this is what is in plan with regards to the federated framework, is that we use Beacon for getting or for querying our datasets. So once we have some variant details which we would like to query, this will be given as a Beacon query and yeah, searched over the Beacon network. And yeah, this will be returned, for example, from a source like EGA. And if we of all the datasets, if we're able to identify the ones of interest, access to that can be given by Elixir AI, which yeah, in principle could be automated, but yeah, we are not quite sure at this point in time. And yeah, the dataset, once we have authentication to download it, we will be storing it in some sort of data staging area, which has an computational environment present for running the PRS. And you know, this will be run using the Nextflow task services. Yeah. So sorry. So that is with regards to the PRS use case. So yeah, I would like to thank Will, Andrea, and Andre, YC, and Leslie from HMGU for helping me out with a lot of queries and giving me ideas during the development of the pipeline, and also Kaur and Nurlan from Tatu, who basically gave me an introduction to Nextflow and also helped me understand the EQ2L pipelines for the federated framework implementation.
Thank you very much, Will and Anshika, for that presentation. So anyone can ask questions now in the Q&A panel. We already have some, so I'll start reading them, and they have been coming during the presentation. So the first one is, on monitoring the use of health data, say if a certain group or individual asks how their data were used, what would you show them?
I think I would, it depends on, obviously the use case that we're doing with that, but for what we're doing, it would be a question of showing people the analysis work that we've done, the analysis plan potentially, and showing people some of the summary statistics from the results from the analysis that their work has been part of, and explaining to them how that has contributed to the understanding, hopefully, or better understanding of the disease.
Thank you. So the next one is, how does PRS use GWAS?
The association files, that the effect sizes that are required to compute the PRS, they are from the GWAS catalog. So they are already present in a lot of data sources already, and yeah, depending on your data set, the population and the phenotype, you can download the GWAS file and use it.
Okay, thank you, Anshika. And then the third question we have is PRS scoring computing on demand or scheduled run across the population?
I don't understand what is on demand. What does it mean in general? So if we have a set of genotype files, I mean, basically we have the genotypes from a given population, we can, and we want to assess certain PRS scores with regards to certain diseases or traits. Yeah, we can, the pipeline can be basically used for that. Yeah.
Okay, and then what are the benefits of EQTL?
I guess Will could answer.
Yeah. Yeah. So, I mean, EQTLs are effectively just doing a GWAS, but you are looking at the expression of a gene as your kind of phenotype of interest rather than, say, something like height or weight. So you're associating the expression of various proteins in the body with variants found in their genome. So the idea is that you can hopefully then link the variants to the gene, because there's always this issue around when we find variants that are associated in the genome, we don't necessarily know which gene they're acting on, because some of the effects can be quite long distanced if they're in promoter regions. And the idea is that looking at EQTLs is you can actually hopefully better show that a change in that variant affects a protein, and then you can link the two more accurately.
Thank you, Will. So we don't have any more questions now, but you can still, there's still a bit of time that you can type your questions there. I'm gonna share my screen for a moment so that we can announce some of the next Seneca events. But yeah, if you have any questions, please write them in the Zoom Q&A, and there's one coming. So is there anything that can be learned from non-health focused international PRS development consortia, such as on educational attainment?
Yeah, I think that, you know, you can do a PRS on anything, but the question is actually how much you're looking at the genetics versus the environment as well. And I say if you're looking at something like educational attainment, there are too many, I think, confounding factors that you would want to take into account with that. It's not something that I have much experience in, but you know, I think it's, you know, I think you could do that. You would learn some things, but I think you have to be very careful at the study level of how you actually would do it.
Thank you, Will. And then we have another one. Not sure I understood. To run this in a federated way, it means that analysis of the data will be made at multiple locations, not just data coming from EGA and centralized on one location using HTS-GET. And then this Nextflow pipeline will need to be deployed at multiple data hosting locations.
Yes, I mean, that's the idea, is that this is kind of the diagram that I showed initially with the analysis that's happening at each cohort level. The idea would be that potentially that you're using the authorization to log into that infrastructure and gain access to the data, and then you can upload the code and run it there, and then the generated result is a set of summary statistics that are then transmissible back to the central analysis part for meta-analysis or combination with other results. So this is the ideal. At the moment, it's very difficult because the unanswered question is who vets the data coming out and back to the researchers. So that's a tricky one to deal with, and I think, you know, this is something that the European health data space, for example, is going to have to deal with, and we're going to have to work out how you can verify that any analysis that's run, the results from that are actually suitably anonymized.
Okay, we get thanks for clarification. So next question is, what would it take for hospitals to use this at scale?
I think for hospitals to use this kind of thing is that they would need the infrastructure to be able to set up some analysis side of things for the data storage as well, and also being able to link into the authorization systems and the beacon network to be able to share the data. That should be possible, but I think it's something that needs time. And certainly from like the EU point of view, is they're looking now to try and centralize this into the health data space, so that there would be one or more kind of data repositories for this sort of health data per country, and that the hospitals wouldn't necessarily need to be the ones responsible for that. They would be able to push their data over to this secure environment.
The next question is more of naive question on EQTL and GWAS. Could just the summary statistics be helpful in discovering new loci related to diseases using PRS?
So, I mean, the idea with the PRS is that you are looking at sort of summary statistics that you're getting through, and the PRS is really developed from the GWAS. So you're running the GWAS, and you're basically taking some people take all the associations, but I think the most powerful ones are where you take everything that shows a significant association. The problem with using them for kind of discovery is differences between ethnic groups, is that variants that are associated in one region of the world aren't necessarily the same regions causing the same disease elsewhere, and although they may be still affecting the same pathways or proteins. So it's fairly difficult to sort of just look at the summary statistics, but I mean, when we're doing GWAS, the summary statistics are what we use for the discovery, and we get 50 to 100 different groups around the world to run that. We combine those, and that's the discovery, and then from that you can develop the PRS.
Well, thank you, Will. Next question is, how do you communicate uncertainty in the output?
I think that's a difficult question. I think you just have to be open and honest with people if you're communicating that there is some uncertainty around the results. I think it depends on your audience. If you're to other scientists, they can potentially understand that results have confidence intervals. If it's to the general public, then you have to be fairly careful in how you phrase it to make sure that any uncertainty is highlighted clearly.
Okay, thank you. Then next question. It seems there are research that suggests cross-thread assortative mating could be inflating genetic correlation estimates, especially on psychiatric conditions and possibly other health conditions. Is there a benefit in terms of neutralizing such effects in the federated setting?
I think you want to take into account as many things that you know about as possible. So this is something that's sort of relatively new in terms of being suggested that it's a potential issue. I think it's something that would potentially be useful to take into account, because I think the idea is that when you're actually trying to look across the different PRSs, that you do need to take into account differing variables as part of the data set. So I haven't looked at it, or even haven't given that much thought, but I think it's something you probably want to, there might be a benefit, yeah.
Okay, thank you, Will. So if there are any more questions, please type them in, and while we wait for that, I'm going to announce the next CINECA webinar. It's already next week, so it's Thursday next week on 9th of February, and it's about data reuse, so ethical, legal, and societal issues in international genomic data access and sharing. So if you are interested in it, you can go to the CINECA website and register there. And we are also organizing a workshop on federated data analysis, which will take place in Cape Town, so in South Africa, on the 26th of February. And again, if you are interested, you can check the CINECA website, or you can also go on the check the QR code here on the screen. So wait, is there any more question coming? I can give you a few seconds more, and otherwise we'll end here. So one more question: does the choice of GWAS study have an effect on the PRS?
Yes, of course. The summary stats that we are using, it has to be in the context of the PRS that we want to compute, and we have to check whether there are no sample overlaps as such, and but otherwise we'll get skewed results because of it. And yeah, I guess the population stratification also has to be taken in mind with regards to what GWAS we have selected to take the effect sizes.
Thank you very much. So thank you very much, Nshika and Will, for the presentations today. And thank you everybody for attending. And yeah, if you want to keep up with the CINECA activities, check our website or follow us on our Twitter channel. The recording of this webinar will be on the website in the next couple of days.